Hi everyone! By now you've watched a lot of videos related to cells. This video is going to do a review of those different topics and give you the highlights just to recap the module. Now it's not going to cover every detail that you have to know related to cells, but it should give you a good summary of those topics. So we're going to review movement across membranes, how tonicity affects that movement, the structures within the cell cycle, cell division, the basic structure of DNA, how that DNA replicates, protein synthesis, or how you read the DNA instructions to make proteins, and then a little bit about stem cells and mutations. So let's get started. So in this table, we can summarize all of the different types of movement across membranes. All of the types of movement that you're looking at here on this screen are passive types of movement. Diffusion, osmosis, filtration, also known as dialysis, and facilitated diffusion are all passive types of movement. Now, what does that mean? If it's passive, then you're not exerting any energy, right? So when we're talking about passive types of movement of molecules, there is no energy source needed. These molecules are naturally going to move down their electrical gradients or down their concentration gradients, either towards something of an opposite charge or to where there's less of that molecule. It's a little bit like a crowd of people in one area of a room. If they keep bumping into each other, eventually they kind of spread out so they're not bumping into each other as often. When we talk about diffusion, we're really talking about just anything that's small enough to move. And that molecule or those molecules are always going to move down their concentration gradient. So they're going to bump around and they're going to move to where there's less of those molecules. When we talk about osmosis, we're specifically talking about the diffusion of water. So this is still a type of diffusion, but now we're talking about water crossing a membrane. Since it is diffusion, again, we're talking about water moving from where there's more water to where there's less water. Dialysis or filtration is similar to osmosis in that it is a type of diffusion. In this case, however, we are talking about the diffusion of solutes that are going across that membrane. So these are small atoms or molecules that are able to move down their concentration gradients across that membrane. Again, because it is diffusion, we are talking about moving down that gradient. Now, facilitated diffusion has the word diffusion in the name, so we again know that this is a passive type of movement and that we will be moving down our concentration gradient. But what we are talking about now are small molecules or charged atoms that maybe can't move across that membrane on their own, because remember, the inside of that cell membrane is nonpolar or repels water or anything else that would be attracted to water, like ions or small polar molecules. So in order to move across the membrane, they need to be facilitated or helped out. So a facilitator helps something out. I am, in essence, a facilitator of your learning, at least hopefully. So in this case, we're going to have some kind of protein channel that acts like a tunnel that these molecules or atoms can move through. Now, we're not carrying those atoms or molecules across the membrane because that would be using energy. So we're just providing a tunnel or a bridge that these things can use to cross that membrane. So in this case, we must have a protein channel. So those are the different types of passive movement. Now let's take a look at active movement. So when we look at the different types of active movement, we're talking about active transport, maybe just using a transport protein, a protein pump, Or we might be talking about moving lots of things or really big things all at once, which would be endocytosis or exocytosis. So all of the ones on this page, if you remember, are all different types of active transport. If you're active, that means you're using energy, right? So that means we are going to have to be using lots of ATP for these processes. Now, if I'm just using a little protein pump to carry stuff across that membrane, then I'm typically carrying really small things, maybe those really small molecules that can't cross on their own. But more often we're carrying ions. Because remember, ions are charged. Water is a little bit charged. So these ions are attracted to the water, which means they're not going to want to go through those tails of the membrane that are non-polar or repel water. In this case, we're going to be moving these atoms or molecules against their gradient. So we're going to go from where there's less to where there's more. Now we start talking about moving more things at once. So in this case, we're talking about endocytosis or exocytosis. Now you'll notice over here on the left, there are 3 different types of endocytosis. There's pinocytosis, phagocytosis, and receptor-mediated endocytosis. Pinocytosis means water, so now we are moving a lot of water and anything that's dissolved in those droplets of water. Sorry about that. And in this case, endo means within or into, so we are moving these droplets of water into the cell. Phagocytosis means cell eating. We are phagocytizing other molecules or particles. We might be engulfing a bacteria or a cell that's no longer functioning properly and we need to break it down. There are lots of different types of phagocytosis. So we are often bringing in large particles or big macromolecules, maybe bacteria or an infected cell. And again, because it's endo, that means we are going into the cell. Receptor-mediated endocytosis, remember, means that we're using receptors or these little mailboxes on the outside of the cell. So we have a bunch of mailboxes on the cell membrane, and when enough molecules bind to those receptors that all these little mailboxes are full, then we're going to kind of bubble in that membrane to carry the mailboxes inside along with the molecules that they've grabbed onto. So in this case, we are bringing into the cell lots of one particular type of molecule. So maybe we are a cell that needs lots of cholesterol molecules to make certain types of hormones. Then we want to have lots of mailboxes for those cholesterol molecules so we can grab a lot of them and then bring them all in at once. So again, we are bringing in lots of the same molecule into the cell. The last type of movement across membranes is exocytosis. If endo means we're coming into the cell, then exo is exiting the cell. So again, we're taking large particles, big macromolecules, maybe a bacteria or another cell that we've broken down, and now it's in kind of a garbage bag cell membrane pocket, a vesicle, and we want to dump it all outside the cell as waste. So now I'm going to be taking lots of those particles or molecules, and I'm going to send them out of the cell. So let's take a look at one particular type of movement that we mentioned, which is osmosis. Now, the tonicity of the solution surrounding a cell drives osmosis or water movement in a particular direction. So let's look at these 3 beakers that we have here, and we have 3 cells inside those beakers. If I have a cell over here on the left that maybe has a concentration of solute inside of, let's say, 9%, and I put that into a solution that also has 9% solute floating around it. So that might be salts, sugars, anything that might be dissolved in that water. That means that if these are the same, the solution out here, this ECF, is isotonic to that cell. Iso means same. Water, remember, wants to move down its concentration gradient. So water wants to move where there's less water. Now in this case, we have the same amount of solute on both sides of the membrane, which means we have the same amount of water on both sides as well. However, our bodies are dynamic. They're constantly changing. Molecules are always moving. So just because there's the same amount of water on both sides of the membrane doesn't mean things stop moving. It just means that the amount of water moving out of the cell is going to be equal to the amount moving in. So we get no net water movement. And our cells are happy in this environment. But let's say, for example, we have a cell with a 9% concentration inside, but I put that into a solution that only has 2% solute. If there's less stuff in that water around the cell, that means there's more room for water, right? In this case, since we say there is less solute out here, we say that this ECF is hypotonic to that cell, and there's more water out there. So then that means that this extra water is going to want to go down its gradient and move into that cell. So we get water going into the cell. If enough water goes into that cell will expand and it potentially could burst, or we say that the cell lyses. So in this case, my cell could lyse or break apart. Now, the way you can think about it is that the water wants to go towards the higher number, the number always representing the solute, but technically it's going where there's less water. In this last situation, if I still have 9% solute inside, and I put it into a solution that has more solute, maybe a 12% concentration of solute, then that means that my ECF, my solution out here, is hypertonic or higher than the solution inside. There's more solute out here. If there's more solute in that solution, then that means there's less room for water. So technically there's more water inside the cell at this point. So the water is going to want to go down its gradient or out of the cell. So we're going to get water going out of the cell. And my cell is going to then shrivel up or crenate. So my cell crenates. Now, again, you could think of it as the water wants to go towards the higher number, if that helps you remember the direction of movement. Now, I'll guarantee there will be tonicity questions on the test. So if you see one, you can use your scrap paper to draw out a little mini beaker and cell and put some numbers on it to help you figure out which way the water would move. Now, let's do a quick recap of our cell structures. So if we look at a typical cell, and let me just say this is not a typical cell because we wouldn't see a cell with a flagellum and cilia. That doesn't really work along with microvilli. But this cell has everything on it. So let's kind of just recap what these different structures do within the cell. So let's just kind of start over here maybe with our cell membrane and just kind of loop our way around. So we know that the cell membrane is going to be kind of border patrol and regulate what moves in and out of the cell, as well as allowing that cell to communicate with other cells. Then we move on to our rough endoplasmic reticulum. The rough ER has these little ribosomes embedded on its membrane, which is why it looks rough. And it allows that endoplasmic reticulum to make proteins that it can then package up in little membrane bubbles and then ship out of the cell. So if I had a cell, for example, that was responsible for making a protein like insulin, then I might need to have more rough ER because I need to make a lot of insulin that I can then send out of the cell to get into the bloodstream. Peroxisomes are going to help break down specific types of toxins, in particular hydrogen peroxide that is produced as a waste product of other processes in the cell. Cilia are going to wave back and forth and help to move stuff across the surface of the cell. So I would find cilia in cells, for example, in the upper respiratory tract to help move those little kind of mucus-laden particles out of the respiratory tract. I can find cilia in my dorsal cavity, helping to kind of move cerebrospinal fluid around the brain and spinal cord. And I can find cilia in the reproductive tract, helping to move the egg down towards the uterus. Microtubules, I'm going to just kind of lump in here with my cytoskeleton. So there's some microtubules there and there. And we also have some in our flagellum over here. These different microtubules and microfilaments, these are all going to provide a framework for the cell, similar to how your skeleton provides a framework for your body. And in fact, vesicles actually move along these filaments or tubules, kind of like roads or train tracks within the cell. And they actually kind of walk their way along those tubules. Then we have lysosomes. We know from the last slide that if water moves into the cell, it can burst or lyse. So to lyse is to break apart. So lysosomes are filled with enzymes that help to break things down. I'm going to have lysosomes where I need to maybe break down food molecules, but I also have lysosomes to break down old organelles, or I can do something called apoptosis, where the lysosomes break open and essentially cause cell suicide and digest or break down the cell from the inside out. So if I have a cell that's not functioning anymore, I can use lysosomes. Then I have smooth ER. Now the smooth endoplasmic reticulum does not have those little bumpy ribosomes on it. So instead of making proteins, now we have a factory that's going to make your lipids and your carbohydrates. So if you have a cell, for example, that needs to produce lots of lipids, like cells in the ovaries that produce lots of steroid hormones, because remember, steroids are a type of lipid, then I might have to have lots of smooth ER so that I can make lots of those steroid hormones. Then we have our Golgi apparatus here that kind of looks like this stack of pancakes. And the Golgi apparatus is going to act a little bit like the post office, and it's going to package up or receive those different molecules that have been made by the endoplasmic reticulum, modify them a bit, repackage them in new vesicles, and then ship them on their way. So it might ship those out of the cell, for example. We have over here microvilli. Now you'll notice the microvilli here are much shorter than my cilia, because microvilli don't move and they don't move stuff across the surface of the cell. Instead, what they do is increase the surface area of that cell so that we can have more contact with the outside. So for example, in your small intestines, you have a lot of food passing through that digestive tube. So imagine if we were to straighten out that tube of your small intestine here, and you've got food kind of traveling down that tube. So you're going to have these cells that are lining that tube that are going to have these little microvilli on them to increase the surface area so there's more contact with that food so that we can pull those important molecules into the bloodstream. Then we have our mitochondria. Now mitochondrion is singular, mitochondria is plural. When I ask people what the mitochondria do, usually the favorite answer is it's the powerhouse of the cell. But what does that actually mean? It's providing the energy or most of the energy for this cell, and specifically it's going to make ATP. So I don't want you to tell me it's the powerhouse of the cell. I do want you to be able to explain to me what that means. So I want you to explain that it's going to actually produce ATP. When we get to the muscular system, we're going to go into the mitochondria and talk about all the specific processes about how they produce lots of ATP. And I save that for the muscular system, partly because you've got a lot of stuff being thrown at you right now, and partly because your muscles utilize a huge amount of ATP in the body, so it makes sense to talk about ATP production there. Then we have these little centrioles here. These centrioles are going to be very important because when your cell needs to divide,
They're going to duplicate, and you're going to get one set that goes to one side of the cell and one set that goes to the other side. Then we're going to break down this nuclear membrane, and then we're going to get little filaments that go from these centrioles to the DNA and are going to pull those chromosomes apart during cell division. So if you were to be able to produce a chemical, for example, that destroys the centrioles of a bacteria but does not harm the centrioles in your body, then you would be able to prevent that bacteria from reproducing. Then we have the flagellum. Now, a flagellum, flagella is plural, but a flagellum is basically a tail for the cell. The only cells that we have in our body that have a flagellum would be sperm cells that help to move that sperm from where it's deposited to where it finds that ovum or that egg. Then we have in the middle here our nucleus. The nucleus has the chromatin inside of it. There are lots of terms that sound very similar when we get into DNA. Chromatin is kind of this loose, messy, tangled ball of DNA that's inside the nucleus, inside that nuclear membrane. But when we get ready to divide the cell, that chromatin condenses, it kind of wraps around little proteins, and it condenses into nice tight little balls or little strands that are easy to separate. So the nucleus is sort of the headquarters or city hall, and it has all of the instructions in it for how to make proteins. Because DNA, remember, is basically just a giant cookbook of protein recipes. The nucleolus inside that nucleus is going to be where we make our RNA or ribosomes. So that's where we produce those ribosomes that then go out and get either embedded on the rough ER or are just floating free in the cytoplasm. The ribosomes are the chefs of the cell, so they are going to actually read the instructions on the DNA or read the recipes and then produce the proteins. So as a quick recap, the cytoplasm is the viscous fluid in the cell. The cytoskeleton provides support, a framework, and roads within the cell. The nucleus is the headquarters and where the DNA or most of the DNA is stored. The nucleolus is going to be where we make ribosomes inside the nucleus. The ribosomes are going to read the DNA recipes and produce proteins. The smooth ER is where we're going to make lipids and carbohydrates. The rough ER is rough because it has ribosomes embedded in the membrane, so that's where we're going to make proteins that we can package. Vesicles act like little membrane taxicabs that can carry stuff around within the cell. Now, a vacuole we didn't talk about yet. That's like a really big vesicle that takes up the majority of the space within the cell, and we would use that to store lots of something inside that cell. So for example, plant cells have big vacuoles that are filled with water. Your fat cells have a vacuole that's filled with lipids for storage. The Golgi apparatus is going to kind of modify and repackage things that are made by the ER. The mitochondria are going to produce ATP. Lysosomes have enzymes in them to digest and break things down. Peroxisomes break down different types of toxins, in particular hydrogen peroxide or H2O2. Centrioles are going to help to pull chromosomes apart during cell division. Cilia help to move things across the surface of the cell. And the flagellum is the tail on a sperm cell that helps to move that sperm to its destination, which is technically called the oocyte, or what you think of as the egg. Now, this is a good example of something where you could take little pieces of paper and write the name of a structure in the cell on each separate piece of paper, and then on other pieces of paper write down the different functions, shuffle them up, and then try to sort and match them together to play kind of a matching game. I would suggest, though, then you get to a point where maybe you just flip over the functions and then try to write out the names of those organelles so that you can spell them as well. Let's take a look now at the cell cycle. You should be able to draw the cell cycle out and walk through the different steps in each phase of the cell cycle. Now, when we look at the cell cycle, this section here with these little skinny pie wedges, this section here
is where I'm doing cell division or mitosis. So this is where we will have prophase, metaphase, anaphase, and telophase. And then kind of toward the end of telophase, we'll actually divide the cytoplasm. So that's called cytokinesis. So that's where you actually pinch that cell in half, and now you have two new cells. We'll get into mitosis in a minute, but most of the cell's life is spent outside of mitosis. Kind of like you probably don't spend most of your life reproducing and producing offspring. You spend your life doing other things as well. So the rest of that cell cycle is called interphase. And that goes all the way from here, all the way around until we start mitosis. Interphase can be broken up into three separate smaller phases. We have G1, which is, we've just divided here. So we're going to be doing a lot of growth and producing different molecules and organelles within the cell that we need to be fully functioning and to get bigger. Once we're done with this growth, we may actually die. We might break apart because either we're no longer needed or maybe we didn't develop properly. So maybe we have some lysosomes that open up and we end up with apoptosis, where that cell breaks apart. Or my cell might become specialized. Or my body might decide that I need more of these different types of cells, so maybe I need to divide. So to get ready to divide, I'm going to go into S phase, which literally stands for synthesis. And what I'm synthesizing here is DNA. So you'll notice that my DNA actually gets copied way before I go into mitosis, because mitosis is way over here in these pi wedges, and synthesis of DNA is over here. And if you think about it, that makes sense because you have thousands and thousands of genes. And if you needed to try to copy all of those or make a photocopy of them while you're actually trying to pull them apart, things wouldn't go so well. So we need to make all those photocopies ahead of time so that they're ready. And then when I go into mitosis, now I'm just going to take these photocopies of the DNA and I'm going to separate them. Then I go into G2, and this is where I'm kind of getting ready for mitosis. So maybe I get a little bit bigger, so I have more cytoplasm to split apart. Maybe I produce some extra organelles that I might need or extra proteins that I might need for that process of cell division. So practice drawing the cell cycle out and the different steps within it. I promise if you just sit down and copy it a couple of times and then try drawing it on your own, 5 minutes, you'll have drawn it 10 times and you'll have it down. So let's now dive a little bit deeper into mitosis. I want to make a couple of quick points here first. When we're talking about mitosis, we're talking about the reproduction of somatic cells. Soma means body, so these are your body's cells. And when we do mitosis, we're literally cloning those cells. We're making exact copies. So you do mitosis when you need to grow, when you need to replace old cells that have been lost, maybe like skin cells that you've sloughed off, or if I have damaged tissue and I need to repair that tissue. Now, meiosis is when you're producing gametes or reproductive cells. So this is when you're going to do sexual reproduction and you need to combine sperm and eggs to make offspring. You'll get into meiosis when you get to reproductive system in the next course. So for this class, we're just focusing on the steps of mitosis for growth, repair, and replacement. And remember, we're copying somatic cells and making clones or exact copies. We're going to talk in a minute about DNA replication and protein synthesis. These can be really complicated topics, and if you go online and start Googling videos, you can go down some pretty deep rabbit holes and start to feel overwhelmed very quickly. There are a lot of enzymes involved, a lot of proteins involved in both DNA replication and in protein synthesis. You do not have to know any of those for this class. Okay, I stress that. You do not need to know the enzymes or proteins involved
in DNA replication or protein synthesis. You just need to know the major steps and the major players. So for example, in protein synthesis, you do need to know the different types of RNA, but you don't need to know the enzymes involved. So let's get into mitosis. What's going to happen when a cell is actually finished G1, it's not specialized, and it's time for it to divide? Well, all of my chromosomes are here in the nucleus. And the first thing we need to do is make copies of them. But remember, we do that way out in S phase. So we should be well done with that by the time we actually get to mitosis. So now we have all this DNA. In fact, we have 2 copies of all of the DNA inside this nuclear membrane, but we're going to need to split those into new cells. So one of the first things that we're going to need to do is get rid of that membrane. So you can see here, we're breaking down that nuclear membrane. We also, remember, said that we need our centrioles here to be on opposite ends of the cell so that they can pull the chromosomes apart. So they're going to start moving to those opposite poles. And this big tangled mess of chromatin in here is going to condense so that it's easy to pull those copies of chromosomes apart. So you'll see here that they kind of look like little X's, because when you look at those up close and they look kind of like X's, these are actually exact copies of each other that are held together in the middle, and then eventually those will get pulled apart. So this first phase, where we're actually getting rid of that nuclear membrane, we're condensing our DNA, and we're moving those centrioles, that is going to be prophase. Now, as a quick little memory tool, the phases of mitosis are PMAT: prophase, metaphase, anaphase, and telophase. Now, this is a continuous process. So it's not like a cell does prophase and then it stops and says, I'm done with prophase. Now I'm going to wait and move on to metaphase. It's a continual process. So if I have a cell and I want you to identify what phase it's in, it's going to be kind of right in the middle of that phase, so it's easy to identify. So up here you can see there's still that membrane bubble around the nucleus. And here you can see the chromosomes are getting darker and more condensed, and you don't see that membrane around them. Then we get to where our chromosomes are all lined up here in the middle. This is metaphase. So think M for the middle. Metaphase is in the middle. Then we're going to take those X's and we're going to pull them apart and pull those copies of the chromosomes apart towards opposite poles and towards those centrioles right here. And that is going to be called anaphase. You can think metaphase, the chromosomes are in the middle, and in anaphase, they're apart. They're getting pulled apart. Then we actually have to do the opposite of what we did in prophase. So if you've memorized kind of the major steps of prophase, where chromosomes condense, the nuclear membrane breaks down, centrioles move to opposite poles, we're going to kind of do the opposite here in telophase. So telophase is the end. So now we're going to reform those nuclear membranes. We're going to untangle that DNA, and then we're going to do something called cytokinesis. Cyto means cell, kinesis is to cut apart. So we're going to literally kind of pinch this cell in half and cut it into 2 identical cells. So let's take the cell cycle that we just learned about and the phases of mitosis here, and let's kind of tie that in with DNA replication and figure out where that fits into the picture. So remember that this is prophase over here. So that's where I'm condensing the chromosomes, breaking apart the membrane around the nucleus, and moving those centrioles. And then I'm going to go through metaphase where the chromosomes line up, anaphase where they split, and then in telophase I'm going to start doing this cytokinesis where I split the cells in half. Then I move into G1, I'm going to do a lot of growth, I'm going to make different organelles. And then let's say I don't do apoptosis, so I don't break apart and I don't become specialized, but instead I get a chemical trigger, a hormone trigger that I need to replicate. I need to copy myself, make a new cell. So I'm going to move into S phase. That's when I'm going to do DNA replication. This is a very complicated process, uses a lot of different enzymes. We're going to keep it very simple. Basically, we're going to unzip that ladder of DNA, And then we're going to use enzymes to tack new nucleotides to that one half over here to make a new half of my ladder. And then on this side, I'm going to make a new half over here. So here down, you can see that the DNA is split, and you can see here we've started adding new nucleotides. So what you need to know here is that if you have, let's say, an adenine, then it's going to want to bond to thymine. And if you have a guanine, it's going to bond to cytosine. So if I were to give you a strand of DNA, like A, T, C, C, T, T, G, you should be able to replicate that for me and tell me that the complementary strand, the other side of that DNA ladder, would end up being T, A, G, G, A, A, C. Simple, right? Just need to pair A with T and G with C. And an easy way to remember which ones pair together is you can just think, since DNA is pretty critical to who you are, that DNA is where it's at. So now let's get into protein synthesis. Now this was a pretty complicated process, but I boiled that down into a drawing. So let's recap that drawing real quick. So the first thing we need to do is have a cell, and we need to have a nucleus within that cell. So this is my nucleus. Inside that nucleus is going to be my DNA. Now, DNA is big. It's a giant cookbook, right? You've got millions of base pairs that you've got to sift through. Imagine having a cookbook with thousands and thousands of recipes in it that you got from your grandmother. Well, there's no way you're going to want to haul out that entire cookbook every time you want to bake a cake. So instead, what you might do is make a photocopy just of the recipe you want, and then take that into the kitchen to actually make that cake. Because it does a couple of things. One, I don't have to haul the whole cookbook with me, the whole DNA molecule, and I'm going to be able to protect that original copy just in case something were to happen. The first thing that I'm going to do is I'm going to take just the section of DNA that I'm interested in. So just that one gene. So this here in red is a gene. And a gene is a recipe for a specific protein. So this is one recipe for just a specific protein that I want. So I'm going to use a bunch of different enzymes, and I'm going to copy that into a single strand of mRNA. So this is my recipe. Well, it's a copy of my recipe. Because it's a photocopy, it doesn't look exactly the same. RNA has uracil instead of thymine, and it's single-stranded instead of double-stranded. So it looks a little bit different, but it has the same information. So now my mRNA is going to move out through little holes in the nuclear membrane, and it's going to find a ribosome. It could be a free ribosome floating in my cytoplasm, or it could be attached to the rough ER. But here's my ribosome, for example. Now the ribosome is rRNA. So the R stands for ribosomal. The M here stands for messenger. So it carries the message. And this is my chef. So let's make my ribosome blue, just so we don't confuse it with our mRNA. And our mRNA is going to find that ribosome, and it's going to feed through it like you're reading a little ticker tape. So I'm going to go along and I'm going to read that mRNA in 3-letter chunks called codons, and they're going to tell me which amino acid I need. So each 3-letter block tells you another ingredient in your recipe. The ribosome, once it's attached to this mRNA, can't run to the pantry to grab the ingredients. So we need somebody to help out. So we're going to get the help of another type of RNA called tRNA. He's a squiggly piece of RNA, kind of shaped a bit like a T. And the tRNA stands for transfer because he's going to transfer the ingredients to the ribosome. And attached to this tRNA is going to be my ingredients for the protein. Do you remember what the ingredients to a protein are from macromolecules or organic molecules? These are amino acids. Each amino acid has its own special tRNA molecule. So if you were working in a kitchen, if we keep with our baking analogy here, there's only one person that can carry butter, and that's all that person can carry is butter. So that tRNA molecule runs over and says, you need butter? Here we go, we got butter. And you say, great. And you read the next 3 letters on the mRNA and it says, I need sugar now. And you're like, oh, I don't do sugar. That's not going to happen. You got to go find that other tRNA molecule. Yeah, that one will get you sugar. So then a different tRNA molecule brings a different amino acid. As we go along, we're going to grab those amino acids and start linking them together. And when we get to the end of that mRNA, we now have a polypeptide chain. It's going to fold in on itself. It may attach to other polypeptide chains, but ultimately that's going to be my protein. Now remember, the reason I like this analogy so well is if you think about using a recipe to bake something, for example, you don't take the recipe, rip it up into pieces and shove it in the cake. That would be weird, right? So my recipe doesn't get used up in this process. It doesn't become part of the protein. It's a completely different type of molecule. It's a nucleic acid. The chef doesn't cut off his fingers and put those in the recipe because gross. That again is a different type of molecule. All of these RNA molecules are basically just working in the kitchen and then making that cake for us. The cake is the protein and it's made out of amino acids. Now, when we look at this whole process, there were 2 main things that happened in here. We had to copy the DNA onto mRNA, and then we had to read that mRNA to build our protein. The first step that we had here, where we read the DNA and copied that into mRNA, so we created a photocopy of the recipe, that's called transcription. And the process where we read the recipe and made the protein is called translation. So you could think of it this way. A scribe is someone who writes things. You have to write a book before you can translate it into another language. So that's a good overview summary of protein synthesis. And just like with the cell cycle, practice drawing this. The first 4 or 5 times you might be copying a lot of it from the picture, if you can read it, that I drew, and then you're going to start being able to do it on your own. Just sit down and spend 15 minutes and just try to get this drawing down, and you'll have the information that you need to answer questions about protein synthesis. Now, let's talk just briefly about stem cells, mutations, and cancer. A stem cell is a cell that can become any kind of cell. There are embryonic stem cells that are only found in that ball of cells on its way to the uterus. So once it implants in the wall of the uterus and starts to develop, we no longer have embryonic stem cells. But those embryonic stem cells can become anything in the body. They can become a neuron, a muscle cell, an epithelial cell, you name it. But you also have what are called adult stem cells. Now, it doesn't mean you only have them as an adult, because once, like I said, that embryo implants in the uterine wall and starts to develop, any stem cells it has are actually adult stem cells at this point. They're more limited in what they can become. So for example, you have stem cells in your bone marrow called hematopoietic stem cells, and those stem cells can only make different types of blood. So when they receive a particular trigger to divide and specialize, then they're going to go down a particular pathway and they might become a red blood cell, a white blood cell, or a platelet. But that hematopoietic stem cell can't become a muscle cell, for example. It doesn't have the ability to do that. Now, what can happen is sometimes there are mistakes in the DNA. And when that happens, you have a mutation. There are 2 really important genes that you have in your cells right now that are normal, healthy genes that help to regulate if and when that cell divides. These are not damaged genes. These are not mistakes. These are genes that you have. I can't stress that enough. So you have oncogenes Oncogenes are going to regulate if you're going to divide. So they'll tell your cell it's time to divide. A tumor suppressing gene is going to tell that cell not to divide. So essentially you have an on switch for division and an off switch for division. If I had a mutation in my oncogene, that could make the cell think it's supposed to divide when it's not supposed to. So the cell starts to divide out of control. Or I could have a mistake or a mutation in my tumor suppressor gene, and a cell starts to divide like it's supposed to, but then nobody flips the off switch. Nobody tells it to stop dividing because there's a mistake in that tumor suppressor gene. So then again, my cell starts to divide out of control. So there's kind of two ways that you can end up with too many cells in a particular area. You either tell it to divide too often, or you don't tell it to stop. What happens is the cells start to divide out of control, and they actually start to induce blood vessel formation to feed them as well. So now you end up with a ball of cells or a mass of cells that has its own blood supply feeding it so that it can keep dividing more and more. If those cells stay in the location where they started and they don't spread somewhere else, then you end up having a tumor. But as soon as that tumor moves somewhere else, it still has the mistakes in its oncogenes or tumor suppressing genes. So it may go somewhere else in the body and start to divide out of control there. So you can see here, because I created a blood supply for myself, this tumor was able to put these mutated cells directly into the bloodstream and they could travel somewhere else. So an example of that would be something like pancreatic cancer. So my father was diagnosed with pancreatic cancer accidentally. They were looking for something else in his liver and found a mass there. Now you might think, oh, they found a mass in the liver, so he had liver cancer. But they could actually look at the cells within that mass, and they found markers in those cells that were specific to pancreatic cells. So because they found those, then they know that the cells originated in the pancreas, then they moved to the liver. So even though technically that mass is in the liver, it's actually pancreatic cancer because that's where the cells originated from. Another example would be melanoma. Melanoma is cancer of the melanocytes, or the cells in your skin that produce pigment. Because there's a blood supply to the skin, those cells in that melanoma can get into the bloodstream and travel somewhere else. And a friend of mine, his wife actually passed away from brain cancer that technically was a melanoma that had traveled to that other area of the body. You can get cancer showing up in different parts of the body because they carry those mutations in their DNA with them when they travel through the blood supply. So just recap of that, we have different types of tumors. If they stay localized, they're benign. If they move somewhere else, now they're cancerous, and we say that they're malignant or they have metastasized. And then there are two types of genes that here it says they cause cancer, and that's true, but they are actually normal genes that you need to regulate your cell cycle. If they are mutated, they cause cancer. So let's add that here. We have two types of genes that cause cancer if mutated. So oncogenes normally tell a cell when to divide. If they get mutated, they potentially can tell it to divide when it shouldn't. And tumor suppressing genes tell the cell to stop dividing. If they are mutated, potentially that can cause a cell to continue to divide when it's not supposed to. Now, the last thing that I want to mention about mutations is that they're not always harmful. Sometimes they don't affect your health at all. Rarely can you see a benefit from them. So typically mutations either have no effect because you have some redundancy in your DNA, or there are sections of DNA that don't actually code for proteins and maybe the mutation is there, or they have a problematic effect, like preventing you from producing a particular protein that you need. An example of that would be Duchenne's muscular dystrophy, where you actually get a mutation in the gene that codes for a particular protein, and you end up with really severe muscle weakness and the muscle cells kind of collapse. Can occasionally have a mutation that is beneficial. So an example of that would be that there's actually a mutation that's been found that provides some protection to people against HIV because it actually affects the receptor that a virus would bind to. We've talked a bit about receptors before when we talked about receptor-mediated endocytosis. So you have all these little like mailboxes on the surface of your cell. You also have little receptors that act kind of like name tags and other kind of labels or binding sites on the outside of the cell. Those proteins could be produced by a mutated gene, causing it to not work properly. In this case, we think about those receptors as mailboxes that something binds to. Well, if you have a receptor on a cell that the HIV virus normally would bind to so that it can infect that cell, but there's a mutation in the gene producing the mailbox, and now your mailbox, instead of having this nice wooden post and this nice little mailbox with a little door to it, maybe you just get the stick and a flat top and your mail just falls out. So we kind of damage the gene, which causes damage to the protein that's produced from that gene. And in this case, means that the virus didn't have the ability to bind to those cells and infect them. So that's it for our review. I know it's a bit longer than our normal videos, but I wanted to kind of give us a good summary of the highlights from this topic because I know it feels a little bit disjunct when you're going through all the videos. If you have any questions, reach out and let me know.